Thank you, Bert and Bram. And I also would like to thank the patient and the patient organizations that are here today. It means a lot that we can actually be in the same room, and I'm really looking forward to fruitful discussion this afternoon. And I also would like to say that I'm also looking for a postdoc. So, but Valerie and I, we work closely together, so maybe we can have a shared postdoc, for example, and you can commute between Lund and Paris. So I'm not going to talk much about the laminin 111 upregulation approach that we have been taking, but instead I'm going to talk about initially some published work and our efforts to better understand LAMA2-CMD pathogenic mechanisms. So already in 2008, we performed a microarray analysis of DY3K muscle. And we followed that up with the proteomic analysis also in DY3K muscle in 2014. And we have also done some microRNA profiling, both in DY3K muscle and also in DY2J muscle and urine, also with the aim to develop new microRNA-related biomarkers. But the overall aim is, of course, to find novel therapeutic targets and strategies. And basically, we also heard from talks yesterday that a majority of the dysregulated genes and proteins in DY3K muscles are involved in metabolism. Here you're looking at there's a pie chart of downregulated proteins in DY3K muscle, and a large extent of these proteins are related to metabolism. And so we have also analyzed human LAMA2-CMD cells and found that they are metabolically impaired. We cultured the myoblasts for 7 days into myotubes, and we performed gene expression analysis just to make sure that they have a typical characteristics of LAMA2-CMD cells, such as the increased apoptosis and also increased proteasome activity. But we have also in these cells analyzed mitochondrial respiration, mitochondrial content, glycolysis, and fatty acid oxidation with the seahorse technology. And this is work that has been published, and we have shown that mitochondrial respiration is significantly reduced both in LAMA2-CMD myoblasts and also in myotubes. And as control cells, you see those in red, we use lay cells. They have a mutation in the SIRF1 gene encoding a critical component of the oxidative phosphorylation chain. But you see here in the green that mitochondrial respiration is reduced in myotubes and also in myoblasts. So overall basal respiration, maximum respiration, and also ATP production is significantly reduced in LAMA2-CMD cells. And we can restore mitochondrial function in culture. We culture these cells then on collagen, and when we add laminin 211 back, mitochondrial function is restored. And we have also found that there are increased ROS levels, reactive oxygen species, in LAMA2-CMD, both in mice and also in patient muscle with different staining techniques. This is work done in collaboration with Valerie. And so altogether, our hypothesis is that there is reduced mitochondrial function in LAMA2-CMD muscle cells. So there's a relationship actually between laminin alpha-2 chain on the outside and the mitochondria in the inside of the cells. And we have also shown that there is bioenergetic imbalance in LAMA2-CMD cells. So we're thinking that skeletal muscle metabolism could actually be a pharmacological target for LAMA2-CMD. So we have done separate treatments with 3 different compounds in DY2J mice. So first we have tested the compound metformin. So that's a widely prescribed drug for type 2 diabetes. And it was also tested together with L-citrulline in a clinical trial in DMD patients, and the results were published. And in that trial, they tested L-citrulline and metformin in altogether 47 DMD patients for 26 weeks. And I also indicated that there was no overall reduction in motor function decline in ambulant patients, but there was a reduction in the motor function decline in a stable subgroup of the patients. So we have treated DY2J mice with different concentrations of metformin daily oral gavage for 4 weeks. Another compound that we tested is NAC. So NAC is an approved antioxidant. So it's been used, for example, in patients with cystic fibrosis. And we have also treated DY2J mice with the treatment regimen that you see there. And the third compound that we tested, it's another antioxidant, vitamin E. And vitamin E deficiency is associated with muscle weakness and myopathy. And we have treated DY2J mice with this regimen. So all of these data have been published. So I'm not going to go through the data in detail, but two of the compounds, metformin and NAC, they both enhance grip strength in DY2J mice. And of course, the grip strength is not back to wild type levels, but there is a significant increase in the grip strength. And of course, these compounds are not expected to completely cure, but they could maybe be used as supportive compounds. And what we have also shown is that metformin improves muscle histology, in particularly in female mice, and we don't know really why that is the case. But we have also shown that NAC reduces central nucleation, and it also decreases the occurrence of apoptosis, inflammation, fibrosis, and also oxidative stress in DY2J mice. And vitamin E reduces central nucleation, inflammation, and also ROS levels in DY2J mice. So to summarize, metformin improves some of the pathological characteristics, then particularly in female mice. NAC improves many of the pathological characteristics, and then vitamin E only a few. So the question is whether these compounds can be potential future supportive treatment for LAMA2-CMD. And this is work done by postdocs, Sibeli, Vahid, and grad student Bernardo, together with Valeria Lamont. But this has all been published, and I really wanted to share some unpublished data with you as well. But bear in mind that this is just work now in progress, and this is work done by Kinga Gavlik, and I think she's online. So she's interested in figuring out the role of other laminin chains in smooth muscle, heart, and skeletal muscle. And this also refers to the discussion that we had today and also yesterday. So what is the role of also of laminin alpha-2 chain in other organs than skeletal muscle? Now, unfortunately, there are, for example, no flox LAMA2 mice. Maybe that we would need that tool to be able to delete LAMA2 in certain tissues only. But such mice are not available yet. But what Kinga has done is that she has generated mice devoid of laminin gamma-1 chain in the cardiovascular system. So she mated flox mice with SM22 cream mice to remove laminin gamma-1 from smooth muscle and also cardiomyocytes. And these mice, they die shortly after birth, so they are much smaller, and they are also much smaller during embryogenesis. And as you can see here on this picture, so they have severe lung defects. So when they are born, they display less airspace, and they also have cardiac defects, as you will see on this next picture. So we have done some synchrotron imaging here, and these knockout hearts, they display, for example, atrioventricular septal defects. And it's also interesting to note that most laminins are actually absent from these hearts. So laminin alpha-2 is almost absent, alpha-4 is reduced, alpha-5 is reduced, beta-1, beta-2, gamma-1, and gamma-2 are also severely reduced. So these hearts hardly have any laminin isoforms present. But then, since these die at birth, they're quite difficult to work with. She has generated another mouse model, a global and inducible mouse model of laminin gamma-1 deficiency. Now, while we did these studies, there was actually another group published this same mouse model, and they actually showed that laminin gamma-1 has important functions in the intestine. And we were able to recapitulate those findings, showing crypt hyperplasia and also dissociation of villi epithelium from mesenchyme. And you can see there on the graph to the left that these mice, they lose weight, or they don't really gain weight, and that is also true for female mice. And we have also done some functional assays with these mice, looking into smooth muscle contractility using the myographs, and you can see that smooth muscle contractility is very much reduced than in, for example, colon and also cardiac artery. And then these mice, they also have a skeletal muscle phenotype. Now, laminin gamma-1 is actually not completely absent. It's reduced in these muscle, but you can see here that there's less muscle, and they also have quite a dystrophic pathology. But this is work ongoing, so we're now in the midst of characterizing these mouse models. And another study that Kinga is also involved in is that, you know, in LAMA2-CMD, the laminin 211 is absent or very much reduced. And we have also heard that there is a compensatory upregulation of laminin 411, and you'll hear more about this. Now, the question is this compensatory upregulation functional at all in LAMA2? So Kinga, she has generated double knockout mice. So she has generated DY3K mice devoid of laminin alpha 4, but they die very early, just shortly after birth. So we have not been able to analyze those thoroughly yet. But she has also generated DY2J mice devoid of laminin alpha 4. And as you can see there in the picture of the double knockout, these are quite dystrophic, much more so than any of the single knockouts alone. But we are also in the midst of analyzing these mice, and maybe Kinga is online and can take some questions if you have. So thank you very much.